Hello, good morning, everyone. Welcome once again to our lecture series. This is Mrs. Ifeoma Obinwa, and I want to welcome you this time around to PAE 481. PAE 481 with the title Insects and Parasites Ecology and Behavior. Insects and Parasite Ecology and Behavior. And I have two topics to be discussing with you. One is parasites as bioindicators of ecosystem health. Another that we have two subtopics. One is parasites as effective indicators of pollutant stress, and then we have parasites as environmental sentinels. So in the second topic, we have measurement of infection with host populations, micro and macro parasites, the frequency, the distribution of parasite numbers per host, the concept of incidence, prevalence, and intensity of infection. So right away, let's go to the first topic. First topic, which is parasites as bioindicators of ecosystem health. Bioindicators include biological processes. Or rather, before I go into that, as the name suggests, bio and indicators. Bio is anything that has life in it, and an indicator is something that is able to indicate or sometimes able to show or reveal a particular, let me say quality or whatever that you're looking for. So bioindicators include biological processes, include also species or communities of organisms that are used to assess the quality of the environment and how it changes over time. So the environment this time could be the land, it could be the water, it could be the air. So these parts of the environment have bioindicators that are able to assess the quality or to indicate the various changes that occur in those parts of the environment. And changes in the environment are often attributed to anthropogenic disturbances. Example, pollution, land use changes, or natural stressors, example drought, late spring freeze, and all of that. Then bioindicator species effectively indicates the condition of the environment because of their moderate tolerance of environmental variability. So these organisms that are able to serve as bioindicators must be effective in tolerating some environmental variability so that they can be able to withstand those changes in the environment and then show the quality of that environment, whether be it on the positive side or on the negative side. All right. Disturbance and stress may positively or negatively affect energy resources such as food and light, and biotic interactions such as competition, predation, and herbivory. That is the interactions among living organisms. And then the physical, we have water velocity, substrate upon which an organism attaches itself, then the use of a refugiate or ability to lay eggs and all of that, or chemical disturbances like the nutrients in the environment. So these environmental changes may increase or decrease growth and reproduction of an organism. So consequently impacting on the size and productivity of the reproduction and interactions of other species. Alright, so generally bioindicators are used to, assess the health of any part of the environment because it gives us the changes. For instance, if in the atmosphere where we have the air, we know that the standard nitrogen gas in the air is about 78%, and then oxygen is about 21%, and carbon dioxide is about 0.03%. Now, if there is any exposure or introduction of some disturbing gases in the air that is able to alter the normal CO2 percentage, which is about 0.03%, it can increase it And when it does, it affects the living organisms that are found there. Okay, it affects the living organisms that are found there. So there are indicators that tell us there is a shift in the normal quality that we are supposed to have. So these organisms or these processes, be them climate change, whatever, or global warming that we see, that tells us that there are dangerous gases in this air, they are all bioindicators. Possibly it could mean that a particular species of organisms used to inhabit this particular area or this particular habitat, and then for some reason they are no longer there, or you now see a few of them. It means there's a problem. Or maybe there are particular organisms that usually are okay with pollution and all that, and then you start seeing them maybe in water. You start seeing them, more of them in that water. It shows that there's a problem, that something is not all right. So they show us, or they indicate the health of any part of the environment. So what are the functions of these bioindicators? Number one, bioindicators monitor the environment. They help us to monitor the environment, the physical and the chemical changes in the environment. And number two, they help us to monitor the ecological processes, the shifts in the ecological processes. Okay? And number three, they help us to monitor biodiversity, introduction of new organisms. Which organism is available, which organism has suddenly increased in its reproduction, which organism has suddenly decreased in its reproduction, which organism do we now have, or which organism was there before and was never again, and all of that. So it tells us the health of the environment, and also it indicates it indicates indirect biotic effects of pollutants when many physical or chemical measurements cannot. That is, sometimes when we carry out chemical measurements or even physical measurements, there are actual pollutants that might not be detected. But with the help of these bioindicators, it shows us the natural thing. It gives us the natural disposition that maybe for this organism to be here means that it's feeding on a particular organism that is normally not supposed to be here. So maybe there's an introduction of a new organism that is not supposed to be here. Okay, then the fifth one is it helps us to test drinking water for the presence of contaminants, because some of these bioindicators, some of these organisms that serve as bioindicators, they absorb some of these contaminants in water because they also live in that environment. They absorb some of them. So when we now test, when we now see some of these organisms in water, it means that actually there are contaminants. And when we see those contaminants in those parasites, it helps us to know that this water has dropped in its quality. And then you can go ahead to do your water tests to actually know the exact thing that has gone wrong. So we have types of bioindicators. Number one is plant indicators. Plant indicators are indicators of the plant species. So plants, we know, are very sensitive tools for prediction of environmental stresses, because whatever that happens around them affects them. If there are no nutrients, they're not going to do well. They begin to wilt or maybe have stunted growth and all that. So marine plants provide valuable information to predict the status of oceanic environment, as they are immobile and they rapidly obtain equilibrium with the natural surroundings. They help us to, gives us valuable information of what happens in the environment and what are responsible. So the presence or absence of some specific plants or other vegetation provides also ample information about the environmental health. So there are plants that are weeds, Okay, so maybe when you start seeing them, it tells you that these plants are actually there to maybe compete with other normal organisms or other maybe plants, compete with them for oxygen, for food, and various nutrients that are available in that environment. So we have organisms like the lichens, though these ones are not parasites now, but they are found on the trunks of trees and rocks, and these lichens are composed of algae and fungi organisms. They react to ecological changes in forests, including changes in the structure of the forest, example the air quality and climate. So when it changes, it affects the organisms. So that's what bioindicators are all about. They respond to changes in the environment, just like responding to stimuli. Okay, they respond to changes, and those changes can either indicate if the environment is healthy or unhealthy, as the case may be. So environmental stress can be indicated by the disappearance of lichens in forest. As caused by changes such as increase in the level of sulfur dioxide or pollutants of sulfur and nitrogen. So you see, when lichen that is supposed to be found in the environment or that normally exist, okay, when they disappear, it means that something has happened, okay, in the quality of air that they are exposed to in that environment. So it tells that the level of sulfur dioxide has become so much, or they are exposed to it, and then also the level of nitrogen. Remember, I told us that the pure air has about 78% of nitrogen. So anything on the increase affects the living organisms. It affects the living organisms. So they can either leave the environment, they can either leave the habitat, or maybe you start seeing them dying. So when you see such thing, it gives us an information or an idea of what could be wrong in the environment. And then the parasites here include Warfare globosa. It's a plant species, and it is it's an important tool in showing cadmium sensitivity, and it also used to determine or indicate cadmium contamination. Okay, cadmium is one of the metals that can be found maybe in water, the heavy metals. So changes in the diversity of species of phytoplanktons, including Iglina clastica, Facus totus, and Trichilion anus, indicate the pollution of marine ecosystems. So these organisms that find themselves in polluted water, what they do is that these agents that cause the pollution will be deposited in the system. So when you come across some of these parasites, and then when you screen them for, you screen them for whatever possible, you can see heavy metals, you can see some other things in them. It shows that those things are actually contained in that water. And then also it tells us that even the organisms that serve as their host in that environment are at risk. They are at risk. Okay. So we also have animal indicators. These are animal species of bioindicators that we have in the environment. So bioindicators of quality, environment, and changes in the environment. Frogs are basically influenced by changes that take place in their freshwater and terrestrial habitats. This makes them important bioindicators. Then the zooplanktons like Alona guttata, excuse me, like the Alona guttata, the Mesocyclops adax, the Cyclops, and the rest of them. They are invertebrates, aquatic invertebrates that serve as bioindicators. They are animal species So they live at the bottom, or we call them bottom feeders. They live at the bottom of the water, and they help to pick up any change in the environment. They pick it up easily. So that they can tell us what happens in the general health of that environment. So okay, then the next type of bioindicators is the microbial indicators. Microbial indicators are microorganisms that serve as bioindicators. So microorganisms, of course, we know that they are health indicators. Aquatic and terrestrial ecosystems. If you screen your blood, you find Plasmodium falciparum is a microorganism. It means you have the disease we call malaria. So microorganisms found anywhere in the aquatic, in the terrestrial environment, in any organism, indicates the presence of maybe a disease. So they are natural health indicators. So due to their abundance, they are easy to test and readily available. So some of them, when exposed to cadmium and benzene contaminants, develop new proteins known as stress proteins that are early warning signs. They are early warning signs of the invasion of the heavy metals or metal contaminants in that environment. It could be water and all that. So microorganisms, for instance, in oceanic biomass, are responsible for the majority of productivity and nutrient cycle in a marine ecosystem. So we cannot underestimate these microorganisms, because in the marine ecosystem, they form the primary production of energy. They introduce the energy they derive from the sun. They introduce the energy in the ecosystem, in the food chain. Okay, so these microorganisms, they have a rapid growth rate, and they react to even low levels of contaminants. And other physicochemical and biological changes. So they help a lot. They can be used in a variety of ways to detect environmental pollutants in water. Okay, so the presence of toxins in water can easily be monitored either by changes in the digestive system of these microbes. So it can be hindered or disturbed by the presence of toxins. Their lives, their existence can be hindered, or it can be affected by the presence of toxins in the environment. So in comparison, to compare these organisms to available traditional tests, the tests are quick to monitor, but however, the tests are limited, and their limitation is that they can only indicate the changes in the organism due to the presence of toxins. So, under the influence of no metal pollution, there's a bacteria we call Vogicella indigofera. It's a bacterium. It produces a blue pigmentation, okay, which is a biomarker of morphological change that has taken place in the environment, and it can be effectively observed. Okay, so when they produce that blue pigmentation in the environment, in the aquatic environment, it shows the presence of heavy metals. And then it goes on to show that this water environment has been polluted heavily by metals, or you can say it has metal pollution, or the invasion of heavy metals and all of that. So that's blue pigmentation, which is produced by this bacterium, is attributed to the relationship between the concentration of chromium and the generation of that pigmentation, attributed to the relationship between the organism and the concentration of chromium. So it leads to the generation of that blue pigmentation by the bacterium. So examples of parasites that are good bioindicators of ecosystem health. Number one, we have the nematodes. Nematodes as bioindicators of soil health. Nematodes are small organisms that belong to the phylum Nematoda. So these nematodes are small organisms belonging to the phylum Nematoda, and they're also called the roundworms. And soil nematodes are ubiquitous multicellular organisms. That means they can be found anywhere under the soil. They inhabit all types of soil, ranging from cultivated to grazing soils or lands to forest soils. So they are everywhere, and they parasitize insects, plants, as well as animals. So their presence can be monitored for changes in their population, or rather they can be monitored for changes in their population. They can also be monitored for changes in their behavior or in their biology or even in their reproduction, etc. And these changes can indicate a problem within their ecosystem. Okay, it could mean that the environment is not favorable for them due to one or two, maybe presence of one or two parameters, or it could be that the environment is favorable, maybe due to their density or their population at the particular habitat. So they are good bioindicator in that they indicate, or they can be used to reflect the structure and function of ecological processes as regards to their response to different soil conditions. So their presence means a lot in the health of the soil because they can be used to indicate the health of the soil, whether they are favorable or whether they are not favorable, depending on their changes. So the nematodes, they are one of the most widely used indicators of soil health because they can survive in any kind of soil, just like I said earlier. So as an indicator of soil health, they possess a large number of positive attributes, which you're going to see now. Some of those attributes are that they are abundant multicellular organisms. They are abundant, which means they can be found in all types of soil. They are everywhere, no matter the environment, no matter the weather conditions. You can see at least a nematode. Okay, so this presence that they have in different types of soil helps us to indicate the health of that soil in different ways. So another quality that they have is that they are one of the important components of the soil biotic community. They are one of the important components of the soil biotic community, and the assessment provides us an insight into the soil's biological processes. Okay, another quality is that they feed on all components of the soil food web, and their population reflects the amount of food available and its diversity. So if if their food is not readily available in their immediate environment, it shows that their reproduction would drop or their population would drop. Okay, and then also based on their feeding habits, nematodes can be grouped into 5 major groups. There are the bacterivores, the fungivores, the plant parasites, the predatory, and the omnivores. So these organisms function at different levels of the food web, okay, providing food for other organisms and also feeding on other debris and all of that. So the nematode community are sensitive to changes in the soil caused by different agro ecosystem management practices. All right. Then we go on to fish parasites as bioindicators of water health. The fish parasites. When we come to water, Of course, we have fishes, both marine water and fresh water habitats. Fish parasites are the most abundant bioindicators that we have in water health. Okay, of course, fishes are vulnerable and they are heavily exposed to pollutants due to their feeding and living in the aquatic ecosystem. And due to their nature, they cannot avoid pollutants or the harmful effects. So whatever happens in the water happens to them. Whatever that is exposed or whatever that is introduced into the water sure gets to them fast enough. So most times it is easy for the parasites to detect the chemical changes or the chemical states of water, because they live in the fishes, and they are also exposed to those pollution or those pollutants. It could be with heavy metals or some other things. So the parasites or the fish parasites help to detect the chemical states of water and is an important indicator for heavy metals pollution in both freshwater and saltwater. So heavy metals can enter the fish by direct absorption from the water through their gills and their skin, or by ingestion of contaminated food. Their food in the water can be contaminated by these heavy metals, and when they consume them, it gets introduced into their body. The same way that when we consume fishes infested or contaminated with these heavy metals, it gets to us. The same way it gets to them through their food in the water that is contaminated already. So heavy metals as well as parasites induce significant damage to the physiology and biochemical processes, and they may induce serious impairment to the physiology and health status of fish. So examples of organisms that are good bioindicators in water environment is the acanthocephalans and the cestodes. They are considered good bioindicators because they are able to accumulate heavy metal concentrations, especially the toxic types, in their tissues. And fortunately for them, these heavy metal concentrations are not able to do them any harm while being in their system. So that is why, while accommodating those concentrations of pollutants, they still live and they still survive. But they can show us what is happening in the wider part of the environment. They can show us that these pollutants exist here, or that these heavy metals exist here. So additionally, we have trematodes of fish. Trematode parasites that infest fishes, they can also accumulate a lower level of some metals in their tissues than the cestodes and acanthocephalans. And then on the other hand, comprehensive evidence has been provided that the presence of nematode larvae in the body cavity of many economically viable fish species may affect the processing of fish, and may also have implications for the public health of these fishes, for the public health, and not just for the fishes, for the larger organisms like man and some other organisms, some other enemies of these fishes, some other organisms that feed on the fishes. Okay, so it has also been reported that heavy metals was higher in intestinal parasites, in cestodes. Heavy metals like lead and cadmium, they are far higher in cestodes and acanthocephalan species, several hundred times in comparison to the fish tissues. So what it's saying is that these parasites accumulate these heavy metals in several thousand times as can be compared to the fish itself, as can be compared to the fish tissues itself, the liver, the kidneys, the gills, and the muscles of the fishes. So the parasites accumulate these pollutants, these stressors in the environment 1,000 times or several thousand times more than the fish tissues. So, in fact, on the other hand, they even help to filter these heavy metals. Okay, so they don't get so much at the fishes. Alright, so the presence of bile acids in the lumen of fish resulted in the formation of organic metallic complexes, which means it's a kind of response. Okay, to the presence of organic metallic complexes that are found in the water environment. So there are examples of these acanthocephalans. These organisms are spiny-headed helminths. They are helminth worms. There are different fish species that we can find these organisms, and these Aspersentis megahynchus, Aspersentis megahynchus. So it's a spiny-headed is a helminth worm. Okay, that is found in Nototena coryceps. That is a kind of fish. And then Acanthocephalus lucei can also be found in the European perch. It's a kind of fish also. So these worms have their host specificity. They have the hosts that are specific to them. And the cestodes, remember, they are flatworms. Okay, cestodes of a genera including Renebotrum, Tetragonocephalum, and Parorigmatobotrum genera. So they are different genera of flatworms that are good bioindicators in water environments, and they are found in fishes. And then we also have nematodes. They are the roundworms. Nematodes are the roundworms. We have different genera like the Botricephalus, we have the Dichylene, we have the Hysterothylacium, we have the Anisakis, we have the Echinocephalus, we have the Ascaris, and then we have the Anisakis simplex metal. Okay, these are the different genera that we can find in water as good bio indicators. And then on the other hand, I want us to look at the contribution of planktons. Planktons may not be parasites, but planktons are good bioindicators of water health because these planktons form the primary productivity in the water environment. Planktons are composed of organisms with chlorophyll. They have chlorophyll, and that is why they can photosynthesize and form the primary productivity in water environment. So there are two types: the phytoplanktons, that is the plant species, and the zooplanktons, that is the animal species. So these planktons, they consist of communities that float along currents and tides in water environment. They are good biomarkers of water quality, and particularly in lake conditions. Okay, so one of the reasons we consider these planktons as being considered in lakes is to monitor the water quality of the lake when there are high centralizations of phosphorus and nitrogen. So this can be indicated by certain reproducing rates of these planktons. So when these parameters are in water, that is when there is high phosphorus and high nitrogen in lake environment, certain planktons begin to reproduce at an increased rate. Okay, so it's an evidence of poor water quality. It's an evidence of poor water quality, and you usually find this in lakes, that is water bodies that are not flowing. They are kind of still, they don't flow. So planktons also can act as both an indicator of water quality and as the main food source for many fish. So it plays a very important role in water environment. It plays a very important role in water environment. So the role of fishes in this whole plankton thing is that they help in maintaining the proper balance of planktons in the pond, and then they also convert the nutrients available in wastewater into a form which is consumable by humans. So fishes convert nutrients available in wastewater. The nutrients available in wastewater could help in multiplication of planktons in their production. So the fishes that feed on these planktons help to convert that energy in a way that can be consumable to humans. So they maintain a balance of planktons in the environment because they feed on them. Okay, so planktons can be termed in two ways. They are useful and they are also harmful with respect to wastewater fed production of fishes. So when we have planktons in a lake, I have a flow chart here that indicates that in lakes where we have the presence of planktons, it can lead to high concentration of nitrogen and phosphorus, and then it leads to high reproduction in planktons, and high reproduction in planktons leads to poor water quality. So when we have poor water quality, it affects living organisms as well. It affects the fishes, it affects other living organisms that can be found in that water environment. So another subtopic is parasites as effective indicators of pollutant stress. As effective indicators of pollutant stress. So pollutant stress is a stress response from what we call stressors in the environment. Stressors are the parameters that cause the stress, and then pollutant stress is a response to the presence of stressors. Like we discussed earlier, stressors can be pollution, can be the presence of heavy metals, can be the presence of other materials that can trigger the release of stress hormones. Okay, so this pollutant stress is as a result of stressor exposure, which triggers some hypothalamic production, okay, and released in the systematic circulation. So the adrenal glands in this case are responsive. They are responsive to the stressors. So this pollutant stress are able to, upon exposure to pollutants, are able to activate the hypothalamic pituitary adrenal axis to the release of stress hormones. And these hormones exposure and activation takes place in the adrenal glands. So their production can lead to various deleterious consequences, and it can lead to increased risk of cardiovascular disease. It can lead to metabolic dysfunction. It can lead to depression and even reduced cognitive function. Okay, and some organisms that have been implicated here as parasite stress. Some organisms or pollutant stress. Some organisms that have been implicated here are organisms of the orders of Diptera, Hemiptera, Trichoptera, and Diptera Chironomidae. So organisms, though they vary from one aquatic environment to another, depending on the biotic index, of the water body. These organisms, especially the macroinvertebrates, we can see them in water bodies. They help to monitor the reproduction or productivity or quality of this environment, and then implicate themselves as pollutant stress. So they show some kind of response as a result of changes in the environment. They show a kind of response as a result of changes in the environment. Changes that could be a form of pollutants or pollution, and then changes that could be in form of maybe some kind of heavy metals that are found in the aquatic environment. Okay. Now we move on to parasites as environmental sentinels. Sentinel species are biological monitors that accumulate a pollutant in their tissues without having a significant adverse effect. So they are able to accumulate whatever pollution that is found in the environment, whether be it in the air or atmosphere, be it in water. They can accumulate the pollutants in their own tissues, and then the pollutants will not have a significant adverse effect on them, and they can carry on with it, but they tell us, or they are used to show what happens in the larger environment. So that's why they are bioindicators. And we know sentinel to be kind of something that gives protection. So somehow they even protect the fishes, for instance, in the water environment. They protect the bigger organisms that serve as their hosts, okay, because they're able to accumulate pollutants in their own tissues. They're able to accumulate heavy metals that can be found in water in many thousand times more than these pollutants can be found in the cells or the tissues of their hosts. Okay, so pollutants we know can exist in different ways. It can exist in different environmental matrices such as water, sediment or suspended particulate matter. Sometimes they can occur in low concentrations, and sometimes they occur in concentrations that are even impossible to detect them using the normal analytical techniques. But then with these organisms that serve as bioindicators, it makes the job easy, because we can indicate the presence of some stressors in their environment. Okay. Some organisms that have been implicated here too, just like we've been saying all along, organisms like the acanthocephalans, okay, organisms like the cestodes, they are good biomonitors, bioindicators, and they serve as sentinels. Okay, they serve as sentinels. And these parasites that can serve as sentinels, they should have the following characteristics. Number one is that they should have a high accumulation potential for a particular pollutant. And also they should have the ability to resist that pollutant. There should be rapid equilibration with the pollution source. Rapid equilibration with the pollution source. That is, there should be a kind of, the distribution should be maybe random. The distribution should be random from the pollution source. Distribution all around the habitat. Then there should be a linear relationship with the pollutants. There should be a linear relationship with the pollutant. That is there should be something that should bring the pollutants and the parasites together. Maybe because of the feeding habits of the fish or bigger organisms, or because of the preference of their hosts in so many things, maybe as regards to reproduction or as regards to feeding. Some organisms, when they want to reproduce, they move away from maybe the crowd to a place where they can have their young ones. So it could be in those choices or in those movements that a relationship can be established between the pollutant and the parasite. And then the sentinels should also be abundant and widely distributed. They should also be easy to identify. They should be big enough in order to deliver sufficient tissues for the chemical analysis. Okay, and then they should be long-lived and easily aged, so that we can use them. To analyze this quality maybe over time, over a year or more, involving different seasons and all that. So that is that for bioindicators. Measurement of infection with host population. Measurement of infection. When there is an infection from a parasite infestation or a disease or whatever, how can it be measured within the host population? That will take us back to parasitism, because when we talk of hosts, we talk of a parasite. We talk of host, we talk of infection, we talk of parasite. So there will not be a host if there is no disease agent, and then if there is no parasite or no vector that is able to move that disease agent, which is a parasite. So parasitism is a relationship between two organisms. One is a parasite and the other is the host. So the parasite is the one that derives nutrients, it benefits from the relationship, but the host does not benefit at all. Rather, it gets harmed in that relationship. So in such a relationship, in an environment, the host now becomes the environment, the immediate environment where the parasite lives. Becomes the immediate environment where the parasite lives, and then the host environment becomes a kind of a jungle for the parasite. Okay, so in this relationship, or for the sake of this relationship, we have two types of parasites. The microparasites and the macroparasites. The microparasites, they are usually small and the unicellular parasites, and they multiply or they reproduce within its vertebrate host. So they reproduce within its host, and then often inside the cells. Example, the protozoan organisms. And then the macro parasites, they are usually large and they are multicellular, and they have no direct relationship within its vertebrate host. Okay, their relationship is usually in the environment of the host itself. So example are the helminths. So based on the location of the parasites, where the parasites can be found, we have two types. We have the ectoparasites and we have the endoparasite. Ectoparasites, they are parasites that live on the body of an organism. Okay, example, the human louse. Then the endoparasites, they are organisms that live within or inside the body of the host. Inside the body of the host. Ectoparasites live on the body of the host. So endoparasite example is the protozoan and helminthic parasites of man. These parasites dwell or live inside the body of the host. So distribution of parasites. As a matter of fact, the ecology of parasites can be divided into two: the macroecology and the microecology. Ecology has to do with the environment and all other components within that environment that affects the organism positively and negatively. So the macroecology is concerned with the relationship between the between the parasite and the exterior environment, whether it is directly or indirectly. So it's concerned with the relationship between the parasite and the exterior environment. Okay. Whereas in microecology is concerned with the relationship between the parasite and its immediate environment that is provided by the host. So in microecology is the relationship between the parasite and its host, the immediate environment within the host. But in macroecology is the relationship between the parasite and the outside environment, even the environment of the host itself, the exterior environment, and not the immediate environment that is called the host. Okay. There are factors that can influence parasite density or parasite distribution. Number one is flora. Flora has to do with plant species around the environment. So we know that vegetation can serve as food for many hosts of parasites, whether it's an intermediate host or is a definite host. Vegetation influences the parasite population because so long as their hosts can at any point or at a point in their lifetime be found in such vegetation, so the parasite might also be in such location waiting for them so that they can carry on their life cycles. Or once the host can be found in such areas or such location, that is a flora. Okay, that is a flora or vegetation. It means that the parasite, whether they come with the parasite to the vegetative area, they have one or two things to do with it. So in such places, you can find parasites. Maybe by the reason of the movement of their host, or maybe maybe they will be in such an environment waiting for their host, maybe an intermediate host, to carry on their life cycle. Okay, so for example, we have some aquatic molluscan hosts or digenetic trematodes. They survive only where plants in the water and deciduous trees on the banks are abundant. So it is only where these deciduous trees are abundant that you can see these mollusks. Okay, you can see this mollusk. So it means that the parasites that they harbor or that they can harbor must be there waiting for them. Okay, must meet them there. There must be a point of meeting. So another factor that can influence parasite density is the fauna. fauna fauna comprise of the animal species. So since parasitism can occur only if two different species of organisms enter the symbiotic relationship, so the presence and the abundance of whole species are of critical importance. So in for instance, the larva of tapeworm Tinea pisciformis can be found in the liver of rabbits, okay, and will develop into adults only when the viscera of infected rabbits are ingested by a carnivorous mammal, commonly called a wild cat. So the presence and abundance of wild rabbits and wild cat are vital to the maintenance of this tapeworm. So the larva of the tapeworm can only find its way to the wild cat because it will first of all find its way to the rabbit, and the rabbit will be fed by another organism known as a wild cat before it can get to it. Okay, distribution of parasite numbers per hosts. What's the reason for the distribution of parasite numbers in every host? Why is it that a particular host can have a lesser amount of infection or infestation of disease, and then another host can have a large infestation? So it is because of the distribution frequency, or what we call the dispersion of parasite within the host population. So this dispersion of parasite within the host population can be divided into three categories. Number one is under dispersion. There is under dispersion. Under dispersion means that few parasites are distributed among large hosts. Okay, so maybe no matter the population of the hosts that have the parasite, there are no matter the population of the hosts, they have few parasites each. They have few parasites living in or on their bodies. They have few parasites parasitizing the host. So they are under dispersed. Okay, and then random dispersion is when the parasites are randomly distributed. When they are randomly distributed, like there's an equal distribution. It's a 50-50 thing. Is it that you're infected or you're not infected? There is a random distribution. And then over dispersion shows that that the rate of infection is high. It shows that the rate of infection is high. It could be a large number of hosts with few parasites. It means that few hosts harbor many parasites rather. So over dispersion means that few hosts harbor many parasites. So it could be as a result of some factors, maybe such as host behavior or aggregation. Some parasites love to live in aggregation. They love to move in colonies, in aggregation of some infective stages of the parasite. So there are some factors that can affect this over dispersion. That is large amount of parasites in a host. Some factors that can affect them could be direct reproduction within the host. If there's a direct reproduction within the host, it means that a host can harbor many parasites. And then if there is spatial heterogeneity in the distribution of infective stages, that is diversities in the infective stages, if it has, let me say, more than a host, if it has like more than a host, it means that the infective stages of the parasite, it means that the infective stages of the parasite can only multiply in their final hosts or the definitive hosts, and multiply in the definitive host. So it has no attachment with the intermediaries. Okay. There are also factors that can cause underdispersion. Underdispersion, where we have a few parasites among large hosts. Okay, what can cause that is maybe parasite mortality, that's the death of parasites, host immunological processes and immunity. If the host immune system is intact, if the host immune system is intact, you can fight the parasite. And then we have the density dependent processes. So now let's look at the frequency of infection. The frequency of infection hangs around the prevalence and the incidence. Of infection. And the prevalence is an indicator of the number of existing cases in a population, as it describes the proportion of individuals who have a particular case measured either at a given point in time or during a specified time period. At a given point in time, we call it the point prevalence. That is what is happening at a particular time. Then at a specified time period, we call it the period prevalence. You can say maybe annually, this is the prevalence of this disease. There is a prevalence of this disease annually in this particular location. Or there is a prevalence of this disease monthly or weekly, or maybe you're reporting today. That is a point prevalence at the point in time. Okay. And the incidence happens to be the recorded new cases of a disease in a particular area. So incidence and prevalence are not the same, in that the incidence is the rate of new cases or new events during a specified period of time. Like you say, today on 1st January 2024, and this is the incidence, this is the infection we have today. But you can say the prevalence of this disease in 2023, in 2024, in 2025 is also and so. So you are now like having an aggregate of the different incidences. That is what we put up in the prevalence to show the rate of infection of a particular disease. So incidence occurs when a new case is diagnosed, and each new case increases the prevalence. So prevalence is like a conglomeration of different incidences. Maybe what happened in January, February, March to December, we put the data together and say, okay, within this year, this is what is happening. Or maybe from the beginning to the end of of the manifestation of a disease, this is the prevalence in this community. This is the prevalence in another community, as the case may be. So what is prevalence? Prevalence is the prevalence of a disease is the total number of population with the disease divided by the population at risk for the disease. Total number of population with the disease, or total number of people with the disease, divided by the population at risk for the disease. For example, if if finding out a day that 60 people in your town out of the total number of 200 in that town have allergy symptoms. Sorry, I don't have a board where I can, show the mathematical workings. But I believe with the literature, if you have the literature, you can be able to relate what I'm saying. So find out today that 60 people in your town out of a total of 200 have allergy symptoms of a particular disease. Okay, the point prevalence of allergy for that particular day, will be the total number with the disease, that is 60, divided by the population at risk for the disease. Because whatever happened in the community, and then the community or the town had 200 people. So 60 were down or showed the allergy symptoms, and then divided by the whole population at risk, because all of them in that town were at risk of the infection, but only 60 at that particular point in time showed the allergy symptoms. So it's going to be 60 people with allergy symptoms divided by 200 people at risk. So that's going to give us 60 divided by 200 is going to give us 0.3. And then when we convert it to percentage, because prevalence is usually shown in percentages, when we convert it to percent, we have 0.3 times 100, which will give us 30%. Okay, to give us 30%. Then the incidence, or sometimes you hear the incidence rate, is a measurement of the rate at which new cases of diseases occur over a particular period of time. Okay. So let's look at the intensity of infection. The intensity of infection is the number or concentration of parasites infecting a host. Okay, it is also the measure of someone's current infection status. So the mean intensity of infection is shown by total number of individuals of a particular parasite species in a sample of a host species divided by the number of infected individuals of the host species in the sample. So how do we measure intensity of infections? Number one, we measure the intensity of infection by direct counting of parasites. That is when we actually come to the parasite survey. Direct counting of parasite. For instance, we can count the cysts, we can count the eggs, and we can count the worms, as the case may be, in samples of stools, of blood, of urine, of sputum from the host. So, for example, in the estimation of malaria parasitemia in thin and thick film or thin and thick blood smears include the parasite count multiplied by the WBC count, that is the white blood cell count or the leukocytes, over 200. That is the standard. Okay, but whichever way it happens, there is a direct counting. You can count the parasites, you can count the eggs, you can count the worms. So for those ones that are maybe too challenging, like the eggs, we can now use, like for malaria, that's why we now see WBC count, because we are counting the blood cells now that are affected, and even the erythrocytes. We count the erythrocytes, the red blood cells, the parasitized red blood cells, over the number of red blood cells that we can see. So with that, we can count the parasite, the eggs. Okay, number 2 is the use of chemotherapeutic agents to expel and count gut helminths in feces, usually intestinal nematodes. We can use drugs to expel intestinal nematodes. Some people can take drugs, that is, some of these drugs that are used, sometimes you hear people say deworming, or maybe there's already a worm infestation, and then you take certain drugs in order to bring out those nematodes or those worms from your stool. So from the stool, you can even count. Then you can even count their numbers. You can see them, you can count them. So there is also, the third one is indirect counting of eggs at cysts in feces. Indirect counting. Indirect because it is not accurate, but we can make a great assumptions from them. Then indirect measurement of the levels of immunological responses to parasite antigens. It means the level of immunological responses, the responses as regards to the immune system, how the immune system responds to the parasite antigen. So it is a measurement of the infestation. It shows if the infestation of the parasite is high. Of course, if it is high, it's going to destroy or going to pull down the immunological responses, the antibodies that are responding to the presence of the antigens. So analysis of prevalence and intensity of infection. Okay, done a bit of that, and then let's go to types of prevalence. So we have the types of prevalence, and we have varying degrees of prevalence that can occur within populations of organisms. And number 1 is endemic prevalence. Endemic prevalence is the maintenance of a relatively constant, persistent but moderate level of disease occurrence. Okay, it's constant, it's persistent, but at a moderate level within a population. And the percentage of infected individuals here is an index of the endemicity of the parasite. For instance, we can say that malaria infections is an endemic, has an endemic prevalence in Africa, or we can say sub-Saharan Africa. Okay, because it is persistent, but then at a moderate level, because it does not destroy a whole lot of the population. And this kind of diseases are predominantly in maybe young people, so to say. Young people. Okay, then the second one, we have hyperendemic prevalence. Hyperendemic prevalence, it denotes to a persistent and also high and continuous level of occurrence. It is continuous, and it can occur in all ages and groups, whether the young age or whether in the young aged or the old aged. So it can occur in all age groups, and it is persistent, that is, it is continuous in the population. And then the third one, we have the epidemic. Epidemics are we usually use epidemics in outbreaks or sudden occurrence of a disease with considerable intensity. So they are usually they usually occur with high infectivity, very high infectivity. That's why we call them outbreak. Suddenly it breaks out. That is what it means. So they have high infectivity, and then there is moderately high pathogenicity. So in a diseased world like an epidemic, we have an example like the time we had Ebola everywhere. There's an Ebola, we call it Ebola epidemic. It just suddenly bursts out or breaks out. So it's an epidemic. But even in the rate of high infectivity, though as at that time it wasn't really high in every part of the world, because it has to do with movement, it has to do with movements of the parasite from one person to another. So when all the measures were put in place, like restrictions, you see restrictions in airports, in seaports, and all that, lack of contact from person to person, it reduced the disease. Okay, and then another one, the fourth one is the sporadic prevalence. Sporadic prevalence is associated with occasional increase or irregular pattern or scattered occurrence of a disease in a small percentage of the population. So it occasionally occurs at irregular intervals and in less members of the population. Then the fifth is the pandemic. And this one involves the existence of regional occurrence. That is, it occurs across regions, it occurs across continents. It affects almost every area, almost all the people in the world. So it is a global epidemic that is widespread. It is widespread, and then spreading all over several countries and continents worldwide. So increased human mobility, freedom of travel, and migration of man and animals have led to the widespread global dissemination of infectious diseases. So without human mobility, some of these diseases can be tracked, maybe to a particular location or a particular community or a particular race. But because these diseases can be spread maybe from person to person, and once there is increased human mobility, once there is freedom of travel, somebody can easily infect another, infect services and all of that, like we experienced during the COVID-19 outbreak. It was a pandemic because it goes across people of different races, people of different continents, and it affected every area or every part of the world. So it's a global thing. And the last is holoendemic. Holoendemic, on its part, indicates a high prevalence among children in a population. There are certain diseases that are peculiar to children. There are certain diseases that are peculiar to different ages in a population: the old age, the young adults, and the rest of them. So in holoendemic prevalence, it has to do with prevalence of diseases that are usually seen among children, and this prevalence decreases as the age group decreases. It decreases as the age group decreases. An example of such diseases like polio and all that, which we have vaccines. A whole lot of them have vaccines because diseases occurring in children at that young age is sure, because their immunity is still building up. They are still building up their immunity. So, but with vaccines, with immunization, we are able to help them to conquer those diseases. So how do we measure prevalence of diseases? We have different parameters for the measurement of prevalence of disease. Number one is the use of questionnaires. A questionnaire for epidemiological study must be simple, methodological, and thorough. A questionnaire is like a document involving that has questions as regards to, or that will give us information about a particular disease from a particular environment or community. If maybe there is an information that there are deaths in a community, people, scientists can wade in, or disease specialists can wade in, ask one or two questions, ask people, and then maybe when they get to families or direct patients, they can use questionnaires, ask a lot of questions that can be able to give them accurate information about the disease, the causative organism, how it affects them, the risk factors in that community. So with questionnaires, you can get information. Questionnaires can be open-ended or closed-ended. It could be a yes or no stuff, or it could be you could give the person you are giving the questionnaire an opportunity to explain in his or her own words, and that one even gives you the open-ended questionnaires, even gives you open-ended questions, even gives you a better chance of getting accurate information, because they will tell you exactly what it is, other than is it this or that, yes or no, that is direct. So another way we can measure prevalence is by direct observations of parasites. Direct observations of parasites or clinical manifestations within the host. So we can do this by physical examination. Physical examination of the host. Example, eyes warm in loasis, nodules in onchocerciasis, or the parasites proper. When we see disease manifestations, or clinical manifestations, we can have a direct observation. We can observe the boils, observe maybe the nodules, as in the case of onchocerciasis. We can observe anything at all, physical examination. So by direct observation, we can say, oh, this is this disease, or this is what is happening to this individual. The number 3 is by laboratory investigation or diagnosis. Laboratory investigation or diagnosis. So it can be undertaken by serological, immunological, and parasitological examinations and evidences. Okay. These are different techniques that we can use to investigate the presence of parasites and subsequently diagnose the problem. So this method can utilize innovative rapid diagnostic kits, which are replacing the examination of vacating and infective stages in urine, feces, and sputum. So by laboratory investigation, we give accurate diagnosis. By parasitological examinations, we look at the parasites. We look at the parasites. We get to them maybe by smears, by making films. We look at them under the microscope, and we see the parasites, and then we can say this parasite is causing this. Okay. Or by serological means, you can use serum, using a lot of reagents that can cause antigen-antibody reactions, so we can see the presence of some parasites. Okay, then the last one is the emission of vacating or infective stages. So the examination and identification of feces, urine, sputum, and blood for eggs, cysts, and larval forms of the parasites. Emission of vacating or infective stages, like some diseases in some parasites, some parasites have more than one host. So in one host can take them through reproduction, that is, take them through the production of eggs, up to they will grow to an infective stage, and then they will leave the parasites. That's what we mean by emission or vacating or infective cities. So they will exit that host, lying in wait for the next host, where they will carry on their life cycle. So the examination or identification of infective stages of parasites can be done with feces, can be done with urine, can be done with sputum. Sometimes we do that macroscopically, and sometimes we do that microscopically. Okay, so they can exit the body. Example, the body of let me say man, through the stool, the feces. That's why we go against open defecation. You can defecate with the presence of some infective stages of parasite. It could be the cyst or whatever, or the larval forms of the parasite, and then somebody can pick up those cysts or larval forms. Okay, or maybe the cysts or larval forms can be in some smaller animals that we feed on. So without cooking them well, like the meat, like snails, like pork meat and beef, sometimes without cooking them well, these larval forms of various parasites, Tinea solum, Tinea sardinata, and the rest of them, can get to human beings as definitive hosts. Okay, I think that is that for the class. That is that for the class. I will be glad to entertain your questions. I'll be glad to maybe know your feedback. Maybe I'll give us some corrections. I know the video has some challenges because I should have a board where we demonstrate some of these things, where we write and show some of these things. But in the subsequent editions we can improve. So tell us what you think about the lecture. Give us, you can send in your question, you can send your feedback, or maybe there's something you want to add. You can get my number through your course rep, and then we'll talk about it so you get prepared for your exams. So your exam, the part of your exam on this 481 will come from, or my part will come from this little piece I've dropped for you right now. So I need you to study them. Just make sure you understand what bio indicators are. You should be able to describe bioindicator or bioindicators using a parasite, possibly an aquatic parasite. Okay, you should be able to know what prevalence is and how you can calculate it. You should be able to know how to measure or describe how to measure intensities of infection and all of that. So I wish you good luck. Thank you so much for watching and listening. God bless you.